Thank you for tuning in to our Young Women in Bio virtual presentations. We're very excited to announce the availability of these online resources through our brand new YWIB online platform, which provides young women across the United States and in Canada free access to our educational and leadership programs and other opportunities in science, technology, engineering, and math. YWIB is launching this new initiative for young women who are curious about STEM and want to learn more but may not live near one of our 13 WIB chapters in cities across the United States and in Montreal. Young Women in Bio gives girls today the inspiration and support they need to become tomorrow's leaders in science, technology, engineering, and math. We strive to provide education and hands-on experiences in STEM, as well as share our passion for all scientific fields. The launch of YWIB Online comes at a time when there is a real need to provide young girls who cannot otherwise attend our events and programs with access to online programming in STEM. This will help with our goal to ignite curiosity and to inspire young women to see a future for themselves in STEM. We believe that by making these online resources available to a broader audience of girls, we can encourage more young women to find a lifelong passion and career in STEM. YWIB is part of Women in Bio with chapters nationwide and in Canada. WIB is a nonprofit volunteer organization that promotes diversity and inclusion for women in the life sciences and beyond. YWIB partners with leading companies, universities, hospitals, and other organizations to host highly engaging educational and motivational programs for young women interested in STEM. In fact, in 1 year, we had more than 70 events across North America, reaching more than 4,500 young women and had the support of over 250 volunteers. With our YWIB online program, we will be able to reach even more young women. We currently have 13 YWIB chapters across the United States and in Canada, which is part of Women in Bio. To learn more, visit www.womeninbio.org/YWIB, with chapters in Atlanta, Greater Boston, Capital Region, Chicago, Greater Montreal, Metro New York, Philadelphia Metro, Pittsburgh, Research Triangle Park, San Francisco Bay Area, Seattle, Southern California, and Texas. Hi there, my name is MVargas, Chair of Young Women in Bio Texas, and I will be presenting COVID-19, how federal agencies respond to emerging health threats via diagnostics, health threats such as SARS-CoV-2 and COVID-19. In addition, what are the responsibilities of government agencies to manage COVID-19, and how important is COVID-19 testing? What is the FDA and their primary role in the COVID-19 pandemic? The U.S. Food and Drug Administration, or FDA, along with other federal, state, local agencies, and public health officials across the country all work together to protect the public health, protection from emerging threats such as SARS-CoV-2, and prepares for and responds to emergencies in the case of our current pandemic. In response to public health emergencies, the FDA releases Emergency Use Authorizations, or EUAs, to help strengthen the nation's public health protections against CBRNs, or chemical, biological, radiological, and nuclear threats by facilitating the availability and use of medical countermeasures needed during public health emergencies. Medical countermeasures, or MCMs, can include biological products such as vaccines, blood products, and antibodies, drugs or medicines such as antimicrobial or antiviral drugs, and devices such as diagnostic tests to identify threat agents and personal protective equipment, or PPEs. MCMs are used to diagnose, prevent, protect from, or treat conditions associated with CBRN threats, like the current emerging infectious diseases such as COVID-19. Why are MCMs very important? Well, having enough medicines and supplies stored can help save the lives of those who need it during a public health emergencies. The FDA Commissioner recently released emergency use authorizations of in vitro diagnostics for the detection or diagnosis of the novel coronavirus. In the event of a CBRN threat, such as COVID-19, the CDC collaborates with states and local jurisdictions to ensure they have plans and processes in place to receive and provide life-saving medicines and supplies. The CDC provides 3 resources: guidance, technical assistance, and evaluation. In guidance, the CDC helps health departments develop and improve preparedness and medical countermeasures, such as in vitro diagnostic tests to detect COVID-19. In technical assistance, when the FDA releases emergency use authorizations, they provide information to ensure health departments are ready to respond to threats, especially clinical labs who are testing for COVID-19. In evaluation, standardized tools to measure the readiness of health departments to get life-saving medicines and supplies to the public, such as testing, vaccines, and PPEs. In addition, any CDC labs that perform clinical testing on human specimens must be certified through Clinical Laboratory Improvement Amendments, or CLIA requirements, and maintain current certification of CLIA compliance. Before a lab can conduct SARS-CoV-2 viral testing, a laboratory must be CLIA certified and meet applicable regulatory requirements. CLIA regulations include federal standards applicable to all United States facilities or sites that test human specimens for health assessment or to diagnose, prevent, or treat disease. Laboratories must adhere to regulatory requirements, must maintain current certification and compliance to test human specimens. CLIA, which is managed by the CDC, provides scientific and technical advice and guidance to the Department of Health and Human Services in clinical laboratory quality improvement and laboratory medicine practice. Currently, there are over 260,000 CLIA labs in the country. Clinical laboratory test systems are assigned a moderate or high complexity category on the basis of 7 criteria given in the CLIA regulations. And all personnel processing SARS-CoV-2 specimen using proper PPEs can be handled in a biosafety laboratory too using standard precautions. Certified CLIA laboratories use either a SARS-CoV-2 molecular or antibody test that have received an approval from the FDA. As I mentioned before, the FDA has authorized emergency use authorizations for both molecular and antibody tests for COVID-19. In YWIB Modular 5, Dr. Julie Wong previously spoke about 2 types of COVID-19 testing. The molecular tests, or nucleic acid amplification tests, also known as NAAT, are used to diagnose the presence of SARS-CoV-2 infections. In contrast, antibody tests can detect antibodies such as IgG, IgA, and IgM from an immune response to SARS-CoV-2. These are also known as serology tests. Whenever possible, laboratories should rely on molecular tests to diagnose the presence of SARS-CoV-2 infections. However, a negative results from molecular testing does not rule out COVID-19 infection. As Dr. Huang previously mentioned, using molecular tests can have some variation in sensitivity that is highly dependent on the stage of the disease. For this reason, negative results should always be interpreted in the context of the exposure and history and symptoms of the patient. Results from antibody testing should not be used as the sole basis to diagnose or exclude SARS-CoV-2 infections or to inform infectious status. Negative results from antibody testing do not rule out SARS-CoV-2 infections, especially those exposed to the virus and are still within the incubation period. It is also very important that antibody tests have been evaluated, and any positive results may be due to past or present infections with a coronavirus other than SARS-CoV-2. If a laboratory initially uses antibody testing for diagnostic purposes, follow-up testing using molecular viral tests should be performed. How are nucleic acid amplification, or NAAT, and antibody tests performed? An NAAT requires respiratory samples from the patient because SARS-CoV-2 is a respiratory virus. The CDC recommends collecting only the upper respiratory nasopharyngeal swabs. Oropharyngeal or lower respiratory secretions are lower priority. However, sputum and bronchoalveolar lavage fluid can be collected if a patient has pneumonia or other lung infections. Samples are then processed and tested for SARS-CoV-2 RNA. The test includes extraction of RNA from the patient specimen, conversion to DNA, and PCR amplification with SARS-CoV-2 specific primers. What does the NAAT testing reveal? It tells us if a patient is actively infected with SARS-CoV-2. They detect the presence of characteristic sequences of SARS-CoV-2 genetic material, or RNA, in respiratory samples of patients. If viral RNA is detected, it suggests that the virus might be present. What to do with the positive test? You should self-isolate, call your doctor if your symptoms worsen, and seek medical help immediately if your conditions become critical. So how are antibody tests performed? The immune system naturally produces proteins called antibodies in response to infection. Antibodies recognize and bind to foreign molecules called antigens on the surface of pathogens, such as SARS-CoV-2. When an antibody binds to an antigen, it seeks to disable the associated pathogen through direct or indirect mechanisms. SARS-CoV-2 specific antigens are mixed with the patient's blood, and if antibodies to SARS-CoV-2 are present in the blood, the two will bind to one another, resulting in a color change that marks the test positive. It is important to recognize that this test measures the amount of antibodies produced in response to SARS-CoV-2 infection and does not test for the presence of the virus itself. It does not indicate if a person is currently infected. What does the antibody test reveal? It tells us if a person has been exposed to a virus or other infecting microorganisms. They detect the presence of antibodies that are produced in response to infection. It will also identify if a person has been exposed to SARS-CoV-2 and developed immunity against SARS-CoV-2 infection. According to the CDC, it typically takes 1 to 2 weeks for a patient to develop antibodies to SARS-CoV-2. Because of this delay, antibody tests are not currently recommended for the diagnosis of acute cases of COVID-19. The US FDA authorized the first COVID-19 diagnostic test with a home collection option. The FDA issued EUAs for both LabCorp and Everlywell Inc. Both are COVID-19 RT-PCR test home collection kit to permit testing of samples self-collected by patients at home. The LabCorp home self-collection kit includes a specific nasal swab for patients to collect their sample in saline transport carrier. Once patients self-swab to collect their nasal sample, they mail their sample in an insulated package to a LabCorp lab for testing. Everlywell Inc. also has a self-collected nasal swab and a saliva sputum sample. However, before purchasing the kit, consumers must complete an eligibility questionnaire developed in accordance with the CDC recommendations. The test does not detect antibodies or immunity. It is designated to determine only active infection with the novel coronavirus that causes COVID-19. To help report positive cases of COVID-19, the CDC recommends those that do not have the capacity to do their own testing and are sending specimens to the CDC collect key information on COVID-19 case patients on PUI, or also known as Person Under Investigation and Case Report Form. This includes demographic, clinical, and epidemiological characteristics, exposure and contact history, and course of clinical illness and care received. The CDC developed the case report form to standardize the reporting of information on COVID-19 cases. These data will help the CDC better understand the virus and its impact on health outcomes, and inform public health responses to prevent further the spread of SARS-CoV-2. So let's pause and review for a quick pop quiz. In a global pandemic, the U.S. Food and Drug Administration releases emergency use authorizations for what purpose? The answer? In response to public health emergencies, the FDA will release emergency use authorizations to help strengthen the nation's public health protections against CBRNs, or chemical, biological, radiological, and nuclear threats by facilitating the availability and use of medical countermeasures needed during public health emergencies. Medical countermeasures can include biological products, drugs, and devices used to diagnose, prevent, protect from, or treat conditions associated with CBRN threats, like SARS-CoV-2. So let's go over important module highlights. The FDA's primary role is to protect the public health, protect from emerging threats, prepares for and responds to emergencies, release EUAs to help against biological threats. The CDC collaborates with state and local jurisdiction to ensure they have plans and processes in place to receive and provide life-saving medicines and supplies. And clinical labs testing for COVID-19 must be CLIA certified when testing human specimens. And the FDA has authorized EUAs for both molecular and antibody tests for COVID-19, and has authorized the first COVID-19 at-home test. I hope you learned the important points of government agencies, especially in the wake of the COVID-19 pandemic. Please continue to check YWIB online to learn more about the evolving science on SARS-CoV-2 and COVID-19. And special thanks to our YWIB national sponsors. Thank you for joining us, and stay safe, everyone. We are all in this together. Look for more virtual events and important information at www.womeninbio.org/page/YWIB online.